Welcome everyone to this week's edition of the Extracellular Vesicle Club. Today we're privileged to hear from Colin Hisey, who's a newly minted assistant professor at Northwestern University. So Colin has had a very interesting journey through the EV world, and he actually helped with, he was directing an EV core facility on a tour of duty that he did down under in New Zealand. And then he came back to the US, did a postdoc at Ohio State, and you're going to hear about some of the work that he did there. And then, as I mentioned, he is now back at Northwestern. So those of you who were at the ICEVX Tech meeting in Baltimore, Maryland in the US just a couple of weeks ago had the opportunity to hear a little bit about what Colin is working on. But today we have a little bit of an extended format, and we're going to learn more about the exciting work that he's doing. So Colin and I had a little interaction on social media yesterday, and I don't know if it was a threat or or a promise to make a picture of my face out of extracellular vesicles. I hope we don't see that today. I told Colin, we don't want to scare people here. We want to make them like EVs, but definitely this is some very interesting work that he is doing in the engineering space with extracellular vesicles. So just a few housekeeping things before we continue. I just want to remind everybody to put your comments and your questions in the chat box. So ask anything you want, make any comments that you want. And then at the end of the presentation, after Colin has finished, we will allow you to unmute yourselves and ask those questions. I'd also like to thank Zach Troyer, who is a postdoctoral fellow in my laboratory at Johns Hopkins University, who's going to be helping with the moderation today. So Zach is going to be the one who takes your questions and invites you to unmute yourself and facilitates that interaction with Colin today. Let's go right into the presentation. I see that Colin's slides are already loaded here. So Colin, thank you so much for addressing the EV Club today. We're looking forward to hearing about your exciting research.
Thank you very much, Ken. Thanks to ICEV for now kind of co-sponsoring the EV Club. I've seen a lot of these. This is my first one, so I'm really excited to be here. So as Ken mentioned, I'm a new assistant professor at Northwestern. I run a very EV-centric lab kind of at the intersection of micro and nanotechnology, EVs, and a bit of AI. And today I'm going to talk about kind of the full story of a technology that I think a lot of you have seen in a few of my recent presentations that we got working maybe 4 or 5 months after the last ICEV Tech, and now I've presented a few times, but this will be the full story, kind of the inspiration behind it, and kind of full circle where we're trying to go with it. One pitch I want to make real quick is that ICEV is starting a mentorship program. So if you're anything like me and you were a young trainee that was a bit overwhelmed by the EV space and these crazy heterogeneous particles, sometimes low abundance, I think and then trying to understand how to progress your career in that space, focusing on these EVs, please look at the mentorship program. Ask your students to sign up. We're not going to tell you how to do your research, but I think it's really good for professional development to have multiple mentors. And just a shout out to any PIs, assistant professors, or otherwise who have built a lab. I'm in the process of it, and it's kind of a miracle that these things work out. But I'm really excited to get the lab started. So I'm going to talk about a project called Light-induced EV and particle adsorption, which we use to do tunable micropatterning of EVs and other particles now, primarily for biomedical applications. So I may call it LEVA. That was the original acronym we came up with, LEVA. So I'm going to use those terms interchangeably. Let's see if my slides progress. Great. So my new lab is at Northwestern's Evanston campus, which you can see on the bottom left there. I have the arrow in purple kind of points pretty close to where my lab is. You can see Chicago out in the background. I kind of mentioned what my lab's going to focus on, and these, of course, it's being done by brilliant PhD students and my postdoc Mohammed, who's still waiting on his visa. But there we are at our first Labsgiving. So it's an exciting time in the lab. I could kind of skip through this pretty quickly. I think most people on the call have an idea about EV basics, so just enjoy the TEM images on the top right. But as an engineer, I think EVs are really interesting just because of their heterogeneity, their presence in all these biofluids, and their seemingly important role in cell-to-cell communication and even organism-to-organism communication, including across kingdoms, which from an engineering perspective, I think is just fascinating that we can likely build tools that help us understand some of these processes. To me, as someone who worked on liquid biopsies during my PhD and back when circulating tumor cells were all the rage, still they're quite popular. Like, well, why do we care about EVs compared to other things in bodily fluids? And largely the reason is their membrane. Their membrane, and now we know the corona are really what make them special, and it allows them to carry these protected, diverse bioactive contents that reflect their cell of origin. And when I got into the field, the focus, at least from my understanding, was largely in liquid biopsies. But of course, now we're trying to fully understand the mechanisms they're involved in. And I think the most recent push, especially over the last 4 or 5 years, has been into the therapeutic space, which I think is really exciting. And even here at Northwestern, we have people like Josh Leonard who are deep in this space. But one of the tricky things about EVs, especially from a micro and nano engineer, essentially, is I could show you this beautiful TEM image of all these different types of EVs. These are from an ovarian cancer bioreactor, but it's quite tricky to know where, what type of EV these are. If this came from a complex biofluid, what's the cell of origin? And we know from Ida Buzas's great review paper, we can visually kind of see that how much heterogeneity there is in this space. And so our goal really is to build engineering tools that help us understand all these differences and how we can use EVs to help improve human health. There's a lot of challenges, so we kind of have an endless amount of opportunities as engineers. You can see the field has just continued to take off. I think 2024 may start to level off a little bit. We'll see. The most recent data I've seen is that it's continued to become more and more popular in 2024, but there are challenges, as many of you know, in production, isolation, characterization, building biomimetic in vitro models, rigor and reproducibility. So if you haven't read the MySEF guidelines, definitely take a look at that. It's another great resource if you're starting to get into the field. And then obviously there are all these other particles. The figure I showed before was just EVs being produced, but if you're using a complex biofluid, you have lipoproteins, viral particles, et cetera. And so kind of an endless amount of work, which is fun as a new professor, that we can kind of dig into all these different problems and try to use micro and nanotechnology to help solve some of them. So our lab kind of works in different areas involving micro nanotech and EVs. So I got into the field building one of the first capture and release chips, which you see on the top right there, for EVs, just functionalizing PDMS microfluidic devices with antibodies. And then we came up with a simple elution method to get them off the chip. We've used conductive nanofibers that have aptamers conjugated to them using gold nanoparticles, and you can apply a potential and you can basically capture and release a selective subpopulation of EVs. We make these crosstalk chips that you can use on plate readers, time-lapse plate readers, where you can culture different cells on each side and the EVs can pass through, but the cells can't. Recently, I've been really interested in matrix-bound EVs, particularly microsomes, but as I'll talk about today, there's a family of these EVs and particles that are attached to surfaces naturally. We can make microfluidic gradients of EVs and see how cells behave within fluidic gradients, and surface enhanced Raman spectroscopy. So if you've heard my talks kind of before this micropatterning stuff got working, it's probably either around bioreactors or surface enhanced Raman. But today I'm going to focus on the bottom left there. And so we use all these tools within the EV field to isolate EVs kind of interchangeably. So we think of it as just a toolbox. Some applications require you to do TFF, some require you to do size exclusion chromatography. Here I'm just showing them because I'll kind of just skip through them later. I think most people on the call are probably familiar with these. But in our lab, we use them interchangeably. We generally just think, what's our starting material? What's our downstream application? And we use them as needed. So the way that I kind of got introduced to microsomes was a bit indirectly, in this whole idea of EV patterning. It was all kind of based around growth surfaces and how they affect cells. Obviously, matrix-bound EVs and microsomes are on surfaces, but initially I was more focused on synthetic growth surfaces and microfabricated polymer growth surfaces. And so if you look in kind of the two most commonly used bioreactor systems, there's obviously, stirred tank bioreactors and others, but these two, the cell line and the fiber cell, that have been adapted from antibody production, if you take a Dremel to them, as I often do, and kind of cut out the growth surface, you see that there are these fibrous growth surfaces. So the cell line has this kind of cross-stitch fibrous mesh, the fiber cell, which is basically a hollow fiber cartridge has these microfibers that run through it. And so I became kind of curious when we started seeing these incredible yields that you can get from bioreactors. What is it about these? Is it the surfaces? Is it a unique fluidic interaction? What is it about these bioreactors that allow cells to produce so many EVs? And as we saw at ISEF Tech, Stephen Jay's talk, was trying to really mechanistically look at why this works this way. And I was kind of doing this a few years ago as well. There are a lot of advantages to these. I'm not going to go too into bioreactors, but as an EV technologist, having an endless amount of EVs is kind of important to us. So we have, a freezer full probably of hundreds of preps still. The great thing about these is you can use FBS in certain chambers and not contaminate your sample. They can be pretty space and time efficient, and you generally get a pretty concentrated prep. The startup is getting them up and running is where it's a bit tricky, just the initial cost and inoculating them. And then it's mostly just pretty simple maintenance to get really a lot of EV yield. They're also opaque, which can be challenging, and they can be a little bit difficult to monitor. But the surfaces are what kind of initially got this project going. And so a great paper by, I think it was Kenneth Doyle, I forget the PI, but they're at NIH, so I would look up the paper, but I became really interested in this kind of dimensionality of growth surfaces and how it affects cell migration. And then when I got to New Zealand, I thought this would be really interesting to see how this affects EV production. So you can see that, in 2D cells look completely different than they do in like what we call a 1D or 3D substrate. So in the bottom right, those two are micro patterned 1D lines essentially that have been etched into a surface, and then the 3D is a surface of a nanofiber mat. And you can see that the morphology completely changes, and I thought that would be interesting to see if the EVs that go into the culture media would also be different. So we came up with this way of patterning basically 150 millimeter Petri dishes. We make a very simple photolithography mask using SU8. So A through D there, we're basically doing photo and soft lithography. We then take the PDMS mold and spin coat polystyrene on it and then stamp it onto a Petri dish, and we end up with these micro tracks on the surface. You can see you get these cool diffraction effects if you hold it up to the light, which just shows a successful pattern transfer, and we can characterize the surfaces. And the nice thing is that it's scalable enough to just barely get a reasonable EV prep. And so we can look at how the cells behave on these. We can see how they, for more tightly packed microtracks, the cells are more aligned. They have these longer aspect ratios. We can see that their actin filaments are aligned with the substrate. We can do fluorescence microscopy and SEM and quantify this. And what we saw is that the more tightly packed these microtracks are, the more EV counts we were getting. So I thought this was really interesting. Mechanistically, we didn't really understand how, but it didn't fully explain the bioreactors. I think the bioreactors we now know are largely a factor of just cell density, but to some degree, that growth surface topography and very likely the fluidic interactions are also having a big effect on the production. And we saw that this kind of correlated with the speed that these, in this case, MDA-MB231 breast cancer, triple negative breast cancer cells were migrating. So I wish I could have the videos up here. I just ran out of time loading them. But we essentially see that more tightly packed microtracks on the surface cause the cells to migrate more, and that correlates at least with their EV production. But germane to the point of microsomes and back to surface-bound EVs, when we put WGA on these surfaces, we started seeing these trails. So if you look, I don't know if you can see my mouse, but if you look on top of this microtrack and on top of this microtrack, which are these raised topographical features, we just kind of by chance saw some of Liu's work. He's at Tsinghua in Beijing, and thought, let's dump some WGA on here and stick it in the confocal and just see what we can see. And we saw some microsomes there. And so this sent me down this path of trying to understand how growth surfaces, surface-bound EVs, including microsomes and cell behavior, kind of all interact. And because I had this experience in fabricating things at the micro and nano scale, it just made sense for me to dive into this. If we go back to Edith's figure here, microsomes are one of many types of ectosomes. They're like these pomegranate-like structures that form at the tips and intersections of retraction fibers as cells migrate. But they're quite tricky to study. The yields are quite low. I encourage everyone on the call to look into Liu's work. about how they initially did some of the proteomics on this. I mean, stacks and stacks and stacks of flasks, and then trypsinizing all these microsomes off the surface just to understand what was in them initially. But they're not new. So I'm a huge fan of Liu's work. He has this paper, Discovery of the Microsome. I would argue that microsomes, I mean, maybe it's not discovery, but they've been observed at least since the 40s. So on the left here is one of the first study of cells using electron microscopy, like period. And I tried to highlight there, hopefully you can see those green arrows, that there are these vesicular structures at the tips and intersections of these retraction fibers. And I think for a long time, people just assumed that this was an artifact of sticking cells onto surfaces, and maybe they weren't important. But they've been observed, through the 40s into the 60s. They're still observed, obviously. And if you talk to people who have done a lot of SEM or TEM, they're like, I always kind of notice those things. And I think people just disregarded them as kind of an artifact. But fortunately, back to Liu. The top left video here I've seen probably 10,000 times at this point, and is really an inspiration for Leva, which we developed to try to mimic these trails that cells leave behind. There's my mouse. So you can see here for a single cell just leaving this trail, these vesicular structures kind of inflate at the tips and intersections of retraction fibers. If you look at this video, what's interesting is another cell can come along and gobble these up. So at first I thought, okay, it's cool that cells are producing these, but do they actually do anything? This video and another on the right there from Alyssa Weaver's group shows that these matrix-bound EVs, microsomes, whatever they are, can be internalized. And so the question became, how do we build an engineered system to study this with really high precision that allow us to quantify these really accurately? Because up to this point, it's largely based on chance. And so it's kind of plating cells. Maybe some are GFP tetraspanin 4, some are M-cherry tetraspanin 4, which is a protein that's enriched in microsomes. But there weren't really any engineered systems to do this with high precision. So here's my favorite TEM image. You can see this beautiful pomegranate-like structure. They're not always packed this full of these small EVs, but here you can see the retraction fiber. So these are kind of characteristic physical properties that we can maybe exploit as engineers to fractionate these EVs more efficiently. Then to the question of whether this is just an artifact, it's not really going down this rabbit hole if it's just an artifact that doesn't exist in vivo. But fortunately, we have these incredible zebrafish models, which I don't do, but I'm a huge fan of, that show in real time that microsomes are being produced, at least in zebrafish live. And then we can also isolate these from serum. So another paper by Liu, I think they used 50 mils of blood, I believe, for this prep. If I remember correctly, you'll have to check the paper, but you can get microsomes out of blood. So not only are they being deposited on surfaces, but they can end up in circulation, meaning there's some probability that they detach regularly. And I thought this is really an interesting problem to get into. So there's a potential they could even be used as liquid biopsy markers. I'm not going to get into that today, but my main goal is just to engineer a platform to study these and other surface-bound and matrix-bound EVs. Another nice thing is they can be labeled pretty easily with WGA. So if there was a paper that came out basically about this, you can either leave WGA kind of in solution and it will label them as they're being deposited by cells, or kind of post-migration, you can dump on the WGA and label them. And maybe your favorite cell lines on here, but the one that stuck out to me was U87MG, which is a glioblastoma cell line. And so I had a history of studying glioblastoma and breast cancer, so you see MDA, MB231s on there. And so we thought, let's try to just build a platform where we've engineered glioblastoma microsomes. That was kind of the initial challenge. Like, can we engineer these trails of glioblastoma microsomes and then study their effects on essentially trailing cells that we then put on the surfaces? So just to review kind of what's known, they're formed via a process, at least microsomes are formed by something called migrocytosis. So it's as a cell migrates, these are left behind. They can be found in blood. I think they've now been found in urine, I believe, but we'll have to look into other bodily fluids. In vitro, if you want to really grow a lot of cell culture flasks, you can isolate them with following some published protocols, basically trypsinize the surfaces after you've given them a decent wash, spin out your cells that are in the trypsin, and then what's left in that trypsin solution is largely microsomes, which you can then do differential centrifugation, and usually a density gradient to fractionate them. The initial proteomics have given us one main protein to target for expressing these fluorescently tetraspan 4, but these other proteins seem to be enriched as well. WGA is really cheap and a great way to label them. And of course, once these were kind of popularized, there's all these papers about their roles in these different diseases. As an engineer, it's a bit tricky for me to make these kinds of claims, but I think it's really promising that they seem to have some kind of role that could be exploited or better understood in from COVID to tumor migration to wound healing. There's even been a cool paper on the bottom left there of like modeling the physics of how these are actually formed, which I find really interesting. And then, yeah, here on the bottom right, I'm just showing kind of one of the more basic isolation methods that have been used. So just top loading on an OptiPrep density gradient, and then you generally get them enriched in these lower fractions after spinning. We're working now on trying to do this a bit more delicately and make sure we're really removing other large EVs of similar size. But because of this pomegranate-like structure, if you do like a back of the envelope calculation, microsomes should be much less dense essentially because they per volume they have much more lipid because of all the small EVs packed inside of them and those retraction fibers. But that's something that we're still working on. And yeah, they've shown that basically they're different from, in this case, they call it exosomes, really just a small EV fraction. So as this is happening, there's a lot of other work being done, which I think is really exciting. So we very quickly realized the platform we were trying to build wasn't just about microsomes. So Marley Dewey, now her whole lab at UC Santa Barbara is working on matrix-bound nanovesicles. But her previous lab, this is one of their main research thrusts. Retractosomes, which they've named, basically when retraction fibers break up, they leave this vesicles on surfaces because you just have a lipid that's broken up and reforms into these smaller vesicles. And Jaewon Shin's group, I believe he's still at UI Chicago, down the street from us, has worked on these matromeres, which are kind of supermere, exomere type particles, non-vesicular particles that are in the ECM. So to what we've actually done, I'll give you kind of the background, this kind of mixture of expertise of micro nano tech, EVs, and trying to understand that intersection and build a platform to study these matrix-bound EVs using that skill set. But there was no platform at the time, so I've tried several things that I'll tell you not to do. But the general idea was if we can pattern EVs and then put cells on top rather than put cells on a plate and then dump EVs on to measure the effect, there may be a completely different kind of internalization mechanism, different functional outcomes if a cell basically gobbles up an EV from a surface versus internalizing it from solution. But this was really, I think, enabled by bioreactors. So to do this kind of project, you need a ton of EVs because a lot of things I tried didn't work. And you can't really treat them as a precious material. So you need to just try something, throw them away, try something, throw them away. A lot of things didn't work. But the two main things we were trying to accomplish, this started back in 2018, was to mimic a trail left by a leading cell, single leading cell. So imagine single cell migration, like that video you saw, and then a trailing cell can come up and gobble these up. But just focused on the trail rather than trying to confine the cell to a certain migration path, although we're working on that too. The other idea was if you have a spheroid that's migrating radially, in theory, as you get farther and farther away from the primary tumor model in my head, or just the spheroid, you have less and less EVs because less and less cells have migrated over that path. So we wanted these discrete kind of microtrack trails as well as gradients. And so I started working on this in 2018. Because my background's in kind of clean room fabrication, I was thinking a lot of like replica molding technologies, so micro contact printing, reversible microfluidics, and trying different ways of getting EVs onto surfaces by directly patterning them and kind of contacting EVs onto surfaces with very controlled pressure or defined geometries. That didn't work. So maybe it will work eventually, but it didn't work for us. And then last year we got it working finally using a technique that I'll describe where we can basically now make these patterns. And this is largely just being in the right place at the right time, joining Eduardo Reategui's lab at Ohio State. But I put the timeline up here for the young scientists in the chat, just to encourage you, if you think you have a good idea, this isn't necessarily a project that how do I describe it? That would work well in kind of the general context of academia, the publish or perish. So you have to find ways to kind of survive and work on these ideas on the side until they get working. And now it's a major thrust of my lab. But for a good 4.5 years there, it wasn't working. But I thought the idea was good enough. Unfortunately, I had really good mentors during that time, Larry Chanley and Sheree Blenkeron in particular. But also Euphemia Long, who I've had probably a dozen conversations about this project to get it working. So in the end, we ended up doing this with several types of EVs, patterning EVs from different sources. We used a lab strain of E. coli, did a basic prep of TFF and size exclusion chromatography. And then from our cell line bioreactors, using a U87, which again is a glioblastoma brain cancer culture, we do differential ultracentrifugation, and then our small EVs we send through size exclusion chromatography. The large EVs we do a density gradient following Liu's protocol, where he isolated them from serum. Looking at time, I might actually skip some of this, but basically put this in here to show that we know what we're doing. We know how to isolate EVs from showing like different fractions from bioreactors versus flasks, but I may skip this. And here we're just showing like different glioblastoma production rates over time. We know how to characterize EVs. We get a lot of preps, and this was really important in developing the project, but not necessarily germane to the main thesis. So I'm just going to skip through this. Another great thing about having all these EVs is that you can test all the different equipment when you're about to start a lab. So this will be out at some point soon, hopefully. I need to get the video drop data in here, but we can basically compare all these different particle counters because we have an essentially limitless amount of EVs. Okay, to the actual technology. So we call it light-induced extracellular vesicle and particle adsorption, or LAVA. And what we've done is basically adapted something called LIMAP, so light-induced molecular adsorption of protein for the EV field. So essentially, you coat a cover slip or some other semi-transparent substrate with PLL. We generally oxygen plasma treat beforehand just to ensure good absorption of the PLL or whatever base layer you're using. We put MPEG SVA on top, so we're basically linking a passivation layer of PEG by targeting amines on the surface on the PLL. And then generally for this protocol, we'll use like a really high concentration photoactivator. You can use a lower concentration one, which we do for the multi-EV patterning, but you put that on top, and then essentially you have this kind of sandwich of PLL, PEG, and photoactivator, and the photoactivator kind of embeds within the PEG, and then we just go along the surface and expose it to UV at different dosages and with high precision using a digital micromirror device. So once you do that, you just kind of give it a quick wash, put your EVs on, and they passively absorb to the areas that you've exposed, and you can iterate this process to pattern different EV populations. So here I'm just showing a resolution test. On the top, we have kind of our micro tracks, and on the bottom, we have some circles. This is real-time exposure, so it's pretty fast. It's about, that window is a few hundred microns by a few hundred microns, I believe, about 200 by 200. And so you can get pretty big surface areas pattern. The reagents aren't that expensive, and because it's non-specific, we were immediately able to pattern bacterial EVs and large and small EVs from our glioblastoma bioreactors. We can do gradients. So here I'm showing a Gaussian on top. In the middle there is a linear gradient, and on the bottom is a logarithmic. And essentially, it just exposes certain regions for longer. And the more UV photons you're pumping through, the higher probability you have that you've cleaved off enough PEG to expose the PLL on the surface. So we can do this dynamically. It's still pretty fast, and we can create these really cool gradients. And so I think first off, you can see that there are just these beautiful gradients of different types of EVs that we can make. We can quantify these and kind of compare relative to our templates what they look like. But I think more broadly at this idea of kind of writing mathematical functions into cellular microenvironments is really interesting in the broader context of AI and how that's affecting biomedical engineering. So not just with EVs, but more broadly, our lab is looking at this idea of kind of just writing mathematical functions for cells and then seeing their behavior. So you can imagine putting cells on these gradients. This is where our lab is headed, understanding how different EVs and different gradients affect different types of cells. Here I'm just showing, this is an EV crowd, so making sure I have dye-only controls in the supplementary material, the preprint, if you want to check, but I think these commercial EV standards are great for something like this. I don't have to convince you that I need a dye-only control, that we get these really nice patterns this is our resolution test, just showing for circles we can get down to about 5 microns in diameter consistently. For micro tracks, roughly 2 microns. We have had some success, but it's really hit or miss getting down, lower than 2 microns, but it is possible. And then again, using the commercial standards from Vesiculab and Sigma, again, showing these really nice gradients. Something we've just kind of got working and are still continuing to develop is optimizing this multi-EV patterning idea. So the initial thought was, if we have 2 different types of EVs right next to each other, maybe like cancer and fibroblast EVs, can we watch a cell kind of make a decision, assuming all other things equal, and can that affect how cells decide where to migrate? This is a bit trickier than expected. One of the PhD students, Shilal, who's co-first author on this, did a lot of work optimizing kind of the blocking and iterating this exposure and deposition of EV step. But we're getting there. You can see on the bottom there, it's obviously not perfect yet, but I think eventually we'll get really, really nice images of multi-EV patterning with different colors, and then we can watch cells kind of select what they internalize and where they go based on the EVs that are present on the surface. And we've kind of adapted this again from multi-protein patterning, where you're patterning like different antibodies right next to each other. One of the more recent results, we got some exomeres from Vanderbilt. And so here we're getting all the way down to 1 micron, but obviously they're so small that we're losing, at least using turf microscopy, that like single EV visualization. But we're able to pattern those as well. So we could compare, non-vesicular particles to vesicles on surfaces and how those affect cell behavior in the future. And the second row there is just the dye-only control, because we had to use like a non-conventional way of labeling these. Here again, using exomeres, we can make these gradients. So we just think it's a nice kind of robust tool that's pretty scalable, pretty high resolution, and I'll get into kind of how it's working. So we did a bit of kinetic study. I'm a chemical engineer, so this is like right up my alley of trying to understand, how particles absorb to surfaces. So here we're just showing large and small EVs, TEM there, and then this experiment, I still can't believe it worked, but like first time we basically patterned the surface. We had a couple extra wells and we got the surface in focus on the turf, and then we moved to a new well and just dumped EVs on, same concentration. And so what we started seeing are basically differential kinetics based on EV size. So this is turf images stacked together basically as fast as we could get the camera to take them. So this gave us this idea to start simulating this effect. So Chiron, who's another great PhD student in Eduardo's lab, basically used measured and reported values of EVs, parameters of EVs, to simulate this effect, and actually got pretty similar results. So if you see on the bottom right there, the red and purple are basically the simulation and experimental results of the large EV binding, and the top is the experimental and simulation results for the small EVs. And what you see are basically right at the beginning, the small EVs absorb really quickly, which you would kind of expect because they have more Brownian motion. And then the large EVs kind of take a little bit longer, but you reach somewhat of an asymptote pretty quickly. And so just for context, initially when we were doing this patterning, before we actually watched it happen, we would leave these incubating for like 10 to 20 minutes. But we know now that within like a minute or two, we've fully saturated the surface. And this is important when we start thinking about scaling up to 96 well plates or multiple well plates and doing more time-lapse experiments. This is a really quick process, which improves our scalability. So we thought it was largely just due to size and increased Brownian motion of the small EVs, but at least with our initial zeta potential measurements, and for this specific prep, I'm not saying small EVs have a more negative zeta potential more broadly, but for these samples at least, the small EVs had a lower zeta potential. And so we think it's a combination within the context of this experiment of size and charge. And so, yeah, the probability increases the smaller the particle that it will hit the positively charged surface, and then the more negatively charged the particle is, the more likely it is to stick. So to validate that, we then made some cationic lipid nanoparticles. This is also some of Chiron's work. You can see on a similarly patterned surface, you'll see the EVs, or these are, sorry, lipid nanoparticles bind and then come off. Um, Basically do a similar analysis, and if we pattern this Gaussian gradient, you'll see that we get kind of like a exclusion effect actually towards the center, where with negatively charged EVs, we get the most binding. The kinetics are kind of flat because they don't really stick. And then the COMSOL simulations also show something similar. So we're pretty confident it's both size and charge. Before we were just kind of confident it was just size. So what are the applications in my last, I guess, 5 to 10 minutes? So one thing we tried, Eduardo was already kind of in the space of single EV fluorescence colocalization studies. I think a lot of people in the EV space are moving towards this, especially with some of the really convenient super res microscopy systems coming out. Generally, this is done kind of just like a single EV ELISA, where you capture certain subpopulations of EVs, maybe you've pre-labeled them with something, or you capture them and then label them with something else. But we know, based on Randy's work and others, this is from Rachel Mazenko's paper, that tetraspannins may not be we know we're not capturing everything, and maybe we're biasing our results a little bit if we just rely on antibody capture for our fluorescence colocalization studies. The great thing about LEVA is that you can pre-label your EVs as long as you clean them up properly. You can then pattern them on surfaces, and you can even pattern them in gradients and titrate them on the surface digitally, and still do your fluorescence colocalization. So the takeaway on the top right image there is that we have two EVs we can look at with turf. We've labeled for CD63 using a Alexaflor 488 anti-CD63 antibody. And you can also, at the same time, use these molecular beacons that target different microRNAs, for example, and see, at a single EV level if there's colocalization, and then try to quantify these latent patterns within these highly heterogeneous particles. And so I think this digital titration idea is going to be useful across a broad range of single EV technologies. Getting them on surfaces is important for TEM and AFM and certain microfluidic applications. And so being able to pattern them very precisely and titrate them on surfaces, I think, is going to be important. So here I'm just showing for large and small EVs, we can do this. We can do it for different cell lines, et cetera. Back to the original inspiration. So this was, can we get brain cancer cells to migrate on brain cancer microsome trails? This was the thought 4 or 5 years ago at this point. Long story short, we can. So this experiment, just seeing this work was really a relief at this point. But you can see here on the left that we have what I'm calling microsomes in quotes, because I haven't done enough validation, I think, to claim that they're microsomes. We'll call them, large EV microtracks, and then we put U87 cells on top of these microtracks, and we can watch them migrate and start to quantify that. So this is still in its infancy, like most of this project, but you can see obviously their migration is very one directional. We are confining them based on the surface chemistry to those large EV trails, but we're working on ways basically of cleaving off any residual PEG so that they can also go on to just the PLL without EVs if they want. Is there anything else here? Move on just for time's sake. So making progress from the initial inspiration, making progress on single EV fluorescence colocalization, and then just by chance I met Sophia Mayon, who's a PhD student in David Wood's lab in chemistry at Ohio State. And like a lot of labs, they're doing a lot of bacteria cultures, and I'm of course like most EV nerds, like, what are you doing with your culture media afterwards? And yeah, fortunately met her by an autoclave one day and thought, this would be kind of cool to try to do some kind of functional study with bacterial EVs. We mentioned at ICEP Tech that I think bacterial EV technologies maybe 5 to 10 years behind basically MSC and cancer EV tech. So here I'm showing on the left, funny enough, like a cell that shouldn't be there, that just somehow made it through tangential flow filtration and size exclusion chromatography and the initial centrifugation step, but just showing like the size comparison of a dried up cell on the TEM grid versus the EVs. Here's some more, EVs. There's some flagella that co-isolate in here that we're trying to get out. Although maybe for the context of this, it's not that important. And if we take those bacterial EVs and we add it to neutrophils, this is something people have shown in the past, but basically if you have neutrophils in solution, they're kind of happy to stay rounded. We're showing here that the dye that we used, again, using WGA didn't upset the neutrophils. But if we put neutrophils in solution with bacterial EVs, they get activated, their morphology gets different. Some of them start to mitose. They definitely don't like being in the presence of bacterial EVs, and we can quantify that. But the great thing about LAVA is we can start to study this phenomenon known as neutrophil swarming. And so people, including Eduardo, have worked on patterning whole bacterial cells or you know, synthetic microparticles that are coated with bacterial antigen. That's been done in the past, and they make these arrays. But with EVs, we can kind of, and with EVs combined with LAVA, we have a different level of scalability and tunability than those platforms have, because those, again, are back to what I was trying to do initially, which is kind of stamping things. So if we make these big arrays of bacterial EVs and we put on blood-derived neutrophils, you can see that we can start to quantify this effect, which is neutrophils, one basically initially contacts the surface, signals to the rest of the neutrophils, and they start to migrate and swarm towards those areas. And we can make these arrays, we could probably make 1,000 by 1,000 array of these. And so we can get this really nice data, quantifying the rate at which these matrix-bound bacterial EVs can affect immune responses. And I think more broadly, start to understand immune responses, not just to bacterial EVs, but EVs generally and synthetic particles. Here we're just showing that, based on the spacing, the 60 micron patterns essentially can recruit more neutrophils, and they do so at slightly different rates depending on the size. We were actually kind of worried initially that a reviewer might not get the bacterial EV angle, and so we were like, let's just try different shapes too, to kind of show that we have this tight control of geometry. And so, it just kind of initially was kind of showing off the technology. So on the top, you see the star of bacterial EVs pattern on a surface, and then the neutrophil response on the bottom, a square. But then we thought, if these are in close proximity, we can start potentially to quantify like differential signaling. And so we've kept the surface area the same. So here what you're going to see on the left is a circle, and then on the right, a star of the exact same surface area, but different perimeter, because the star obviously has these protrusions. And we noticed something interesting. So initially they start swarming evenly, and then after a few minutes, there's this differential effect where the star starts to recruit a lot more neutrophils. We can quantify this by measuring chemotactic indices. You can see that, right after the initial swarming begins, they start, preferentially moving to the right. And so, yeah, we're not entirely sure where we're going to take this. I'm very new to amino engineering space, but I'm very happy to hear ideas from people. But we think that this could be a really useful tool for like the scalable study of neutrophils. And every time I've shown this, someone talks to me about macrophages. So now I'm learning about macrophages and other immune cells. So I think I'm okay on time. Yeah, I'll kind of finish up. I think growth surfaces, my interest in growth surfaces ultimately led to EVs on growth surfaces. And fortunately, I think just by nature, there's these microsomes and other EVs that are naturally produced, which really help justify pushing this technology over the years. Yeah, LAVA, which is the process I showed you of basically selectively cleaving PEG and exposing PLL at high resolution on surfaces. It's high resolution down to roughly single micron, reliably more around 2 microns for now, but I think that'll get better. It's really tunable. As you can see, we can change the shape dynamically. I can pattern Ken's face, which now I have to do after this presentation, so be on the lookout for that. It's pretty rapid. We showed with our kinetic studies that we can, the EVs absorb within 2 minutes roughly. We're saturating the surface as long as we have a high enough concentration in our prep. And it's really scalable. So we're already thinking, how do we do high throughput screening? If these are important in cancer or other disease states, how do we build models that we can do, robotic pipetting and get all this done in a really scalable way? It's size and charge dependent, so we'll have to really think about changing out chemistries if we want to do this consistently with like cationic particles, for instance. I showed 3 really quick kind of applications that we're expanding on. Eduardo's continuing to work on this too. I think there's, we have pages of ideas that we could expand this into, but what I showed today is single EV fluorescence colocalization, just a proof of concept. Again, cell migration on these microsome mimetic EV trails. We need to validate that they're microsomes, but I think the hard part is done of getting the technology, and now it's more about EV production and fractionation and validation, and then E. coli EV mediated neutrophil swarming, which I think kind of came out of left field. I had no idea that this technology would lead there. But as with a lot of EV technologies, you solve like a really tricky technical problem, and it can lead to things that are completely unexpected. This has happened with my Raman work and some of the AI stuff we did, had applications in other spaces, and now I think this should affect a lot of other um, spaces within EV and particle research generally. So in the future, I'm not going to give away all my ideas today, but basically, there's an obvious, these are really amenable to including within microfluidic devices, which I have a background in. So I think that'll be really interesting to start incorporating these multiple physical parameters of a microsystem, including micropattern EVs, 3D patterning within hydrogels and other 3D substrates, or kind of 2.5D like nanofiber type substrates. Obviously fractioning the different ideas and measuring selective internalization, decision making of cells, building out virtual cells, et cetera. And then, of course, the chemistry is pretty interchangeable. So you can model different systems depending on whatever disease you study or system you study. You could start to build out biomimetic systems incorporating the EVs that are relevant to your system. The main thing is to be really careful, because much with akin to circulating EVs or EVs in solution, if you don't clean up your prep properly and then you try to claim some functional effect. My fear with this technology is that people will have dirty preps, they'll pattern them and then not realize that they're patterning a bunch of co-isolated protein or something, and then they'll see an effect and go, ah, it's EVs. So it's in parallel to other issues we've had. I think maybe Ken and I should talk along with some of the other researchers of getting some of this stuff in the MySF guidelines, because I could see it getting out of hand if there are serious functional effects of these types of EVs. A few nice images just to finish up. Of course, everyone loves Edith's image, but here it is made out of EVs. I think a lot of you have seen this at this point. Here's a cross section. Typically I pulled it from a random. I think Shalal actually found this one. It's a random EV cross section diagram that you've seen, but there it is made out of EVs, and you can see at certain points we're getting down to single EV resolution on our tetraspanin, on our lipid bilayer here, close to single EV resolution. But at the same time, having the scalability of making these, this one's probably 500 micron by 500 micron patterns with pretty high fidelity. And of course, the Hisey lab. And speaking of the Hisey lab, I have to thank all my new students, my postdoc Mohammed, who's going to be joining us soon. This wouldn't have been possible without support at Ohio State from the Legacy postdoc program, from the Center for Cancer Engineering there that funded the bioreactor work and the initial micropatterning work, Eduardo Reatigi's group, and also Derek Hansford there, Salvandir and Carapaya, and Zach Schultz was actually my primary K99 mentor in OSU chemistry. And last but not least, thank you to the NIH for funding my K99. And I think we have a little over what I expected, but we have 10 minutes left. Hopefully that's okay.
Yeah,
there's some EV baked goods, 3D printed EV at the end there, but I'm happy to take any questions in time remaining. Thank you.
Thank you, Colin. That was a really, really interesting talk. A lot of really cool technologies there. So just to all the audience, again, you can enter questions into the chat, and what I'm going to do, I'm going to allow screen sharing and unmuting. And I'm going to go question by question and call on you, and you can unmute your mic, you can turn on your video and ask your question to Colin. I'm going to start though by first asking a question of my own, which relates to EV collection. I was a little curious if you've looked at how different methods of EV collection impact the ability of EVs to be patterned onto the surfaces, and do you see differences in if EVs were collected by, let's say, like ultracentrifugation versus a filtration method? Do you see better patterning?
We haven't looked at that, but I think it's something, if we step back and really look at it as an optimization problem with a very engineering perspective, I think that's something we have to do. Fortunately, we were comfortable with all those techniques. In this case, I tried to stick to kind of what are the standards that people use. So it's a standard bacterial EV isolation workflow, really standard like bioreactor to small EV, bioreactor to large EV. The one very specific one I followed that was maybe a little unique was following the microsome isolation protocol. But yeah, depending on how you prepare them, or even stepping another level back, how they're produced, affects their corona, and the corona is going to be the thing that affects their binding capacity and affinity. You can get very in the weeds going down that path. Our process at this point has been like engineer the surface and just saturate it with EVs. It's a privilege of having a lot of bioreactor preps around. But I do think, you know, depending on what's on the corona, or if we're even thinking like if you shave off the corona, does it still bind the same way? If you just, you know, enzymatically cleave off things on the surface, yeah, all, and then does that then affect the cell internalization? So there's this multi-parametric problem there, but I think you're thinking exactly how we're thinking. Isolation will matter, not to mention the level of contaminants that you'll have in your prep. So for now, we're going to stick to kind of conventional stuff, but depending on what direction my PhD student wants to go, we might get into that nitty gritty of how isolation affects the kinetics. It's a very like chemical engineer problem, which I like, but I don't know if they'll be interested enough.
All right, thank you for the great answer. Okay, so one of the first questions I see in the chat is from Anand Sasidaran. Sorry if I mispronounced your name, but feel free to unmute yourself and ask your question.
Hey, how's it going?
Hey, Colin, good. How
Good
you
to
doing,
see
man?
you.
Good to see you
Good
too.
doing
Great
well,
talk,
doing
by the
well.
way. I really loved it. So
Thank you.
you kind of brushed on like one of my first question about materials used for this particular purpose. So have you tried like if your goal is to also enhance EV production, have you tried these bioblocks and other like other 3D culture situations where you can grow cells in a bulk in a small contained location and make them to produce more EVs? And also, yeah, I'll stick to first question. Then I have a follow-up question, but yeah, I'll let you answer this one first.
So we haven't used those yet. Actually, the ICEF Tech 2 weeks ago now was the first time I'd heard of those. I think Marley Dewey is using them to try to produce her, what does she call them? Matrix-bound nanovesicles. So I think they would be really useful in the microsome world. To date, I've only stuck to what I've used in the past. I'm really comfortable with the cell line bioreactors, but I think we may go down the route of trying those bioblocks, you called them. I need to write that down.
Yeah, I'm like, they're different companies. They make different kind of things. So
Can you message me that
okay.
product info?
Yeah, I'll send you
Yeah.
that. I'll send the details after this. And another thing is that so the material what you use to grow your cells, like the one you make the imprint on. So how so if you change from polystyrene to something else, which is a little more flexible rather than like more hard. So does that actually affect in your production of EVs? That have you looked into that? Not just production, but also the integrity and the molecular information what they have.
So we haven't gotten there, but that's next up. So I've met a collaborator here recently that's electrospinning nanofibers and an expert in that space. And so I think moving into that 2.5D, 3D space is where we want to take this. I'm a bit hesitant to do too much of that before I fully define whether these are functionally important. So there's still this like underlying question of whether this is even worth putting a ton of resources in. So I think first trying to answer the biology question of like how important is it when these things get internalized has to be step one. But then, you know, I want to take it as far as I can on the engineering side, but we've basically only stuck to like cover slips and standard like 98 well plate surfaces so far, but
Yeah.
we're already looking at hydrogels, nanofibers, trying to get them into more biomimetic like surfaces with topography and stiffness and things like that.
And how
I
do
think
you
I answered your question.
Yeah,
So,
you did. And how do you take off the microsomes from the dishes and put it into your system?
so what I was showing is I kind of skipped over it a bit, but I think our dynamic bioreactors are releasing microsomes into the conditioned media. And I followed an isolation protocol to get them out of serum, which is also full of other large EVs. So I have to validate that, but I think from those bioreactors, you can get at least some fraction of microsomes. But if you're doing a conventional culture, which is kind of the gold standard for getting microsomes, you use trypsin. You grow them on put a little fibronectin on petri dishes or flasks. You grow 50 to 100 of them. I think the first prep they did 100 150 millimeter petri dishes. You grow the cells for like 24 to 48 hours, I believe, and then trypsinize everything, spin out your cells from the trypsin, and then you're left within the trypsin of microsomes, and then you try to like very gently get them out of the trypsin using a density gradient. I think he does 20,000 G spins for the initial pellet, and then you layer it in a density gradient and get them out. So there is a conventional way to get these from all kinds of cells. I think for what we're trying to do, we kind of have to find a high density bioreactor way, or it's just too cumbersome to like scale this up the way we want. So hopefully we can produce them from bioreactors long term.
Cool, man. Thank you. Thanks a lot. I'll send
Good
you
to
the
see
details.
you. Okay.
Later, Colin. Yeah.
All right. I also see a question from Ramon Castellanos Sanchez. Feel free to unmute yourself and ask your question.
Hey, Colin, great talk.
Hey, thank you.
I was just very intrigued about the work with immuno cells. I was just like actually thinking about what's the endpoint or what are the endpoints that you have seen on printing EVs, especially because you mentioned you have like an idea of a controlled EVs, like let's say fibroblast derived versus cancer, and then maybe just put in the immune cells. I wanted to have your take on some translational applications that this make great. So yeah, that's pretty much what I wanted to hear.
So I am so new to the immune world generally that it's very premature for me to comment on anything translational. Just being completely transparent. I think for now the platform is a means of trying to understand like fundamental biology questions of how neutrophils interact with EVs on surfaces, things that can be quite tricky to study in vivo. But I think it will largely be up to my collaborators to kind of help me understand if there's any value in the first place. For now, it's just kind of like a cool thing that we showed works and that there's some geometry somehow affects neutrophil signaling. But in the long term, I think it'll have to be me either learning a lot more or having postdocs that know a lot more or finding collaborators that can give me like the broader context. Because every time I try to learn more about the immune system, I'm just kind of perplexed by how interactive all these different types of cells are with each other and how this could fit into that. Yeah, it's way too early for me to comment on translation, but I think maybe, an obvious thing would be people are trying to make vaccines and stuff out of a lot of different EVs. Maybe this is a way of like screening inflammatory responses to synthetic particles or other particles you're trying to engineer. Maybe that's the first way instead of having, maybe before animal models or in parallel with animal models, trying to understand inflammatory responses. I'm not sure though. I'm very early in it. I'm very far on like the engineering side of biomedical engineering. And so it always takes a while for me to think through like, where could this lead? Currently it's largely, I have this really cool technology. Do you have an application? And trying to think through while also building out the tech of where it could lead. So if you have any input, I'm very happy to hear your.
Oh no, I was just wondering if you have done that experiment with immune cells where you have cancer-derived EVs and then some control like fibroblast derived, and if there's any preference for the cells.
Not yet, but I think that's one of the first things we'll try. So far we've literally only used bacterial EVs, which have a very obvious, antigen that's presenting to the neutrophils. I think now that the technology is a bit more developed, we can get into more of those subtle differences like cancer versus fibroblasts versus endothelial cells or something. So that's where we're probably heading first. But then again, it's like, okay, if we show there's a difference, where is the translational aspect? I'm not entirely sure. It's still, it's a good basic science question we can answer and maybe leads to something like all these technologies seem to. So yeah, short answer is I don't know.
Okay. Well, good luck to you.
Thank you.
All right. Natalie Turner has a question about EV lipoprotein interaction.
Hey.
Hey, Colin, great talk.
Thank you.
So I really love this, and I hope I actually understood it properly. I feel like I've seen you talk on it like 3 times because I just, engineering is not my background, but I saw, so you basically used different colors for different antibodies at one point, like CD9 and CD81 or CD63, something like that. So could you do the same with like EV versus non-EV particles? Because that's what I love. I love this, like not just EVs, but the interaction that they have with these other non-EV particles. Have you done those experiments already or thinking about them?
Yeah, it's something we've already kind of dabbled in. Eduardo's research group has looked at, I don't know if they were just labeling or capturing with like ApoA, ApoB, and then trying to kind of do co-capture and within the same field of view. It's something we thought about. I haven't fully like fleshed out any of those ideas, but I think there's a straightforward like quantitative approach there of just like how pure is your sample. But we haven't gone down that path at all. But if assuming if you want to do selective antibody capture or even somehow exploit differential charge to capture these things, you could like label them both in solution and then pattern them and have a quick quantitation. But it's not something I've worked on personally yet. I think Eduardo's group may be expanding more into that space.
Thanks. Good talk.
Thank you.
All right, I see one more question from Mahmoud Hussein. Do you have a mic?
Yep, thank you, Dr. Hisey. I'm a master's student at North Carolina A&T State University, and we're working on using elastin-like polypeptides for applying selective capture of EVs. I was just curious if you have considered using aptamers in the micropatterns for better, enhanced attachment.
Yeah, we've thought about it. So far, the main approach for this specific way of using this technique has been to focus on label-free capture. But you can do like we showed a little bit of multiple antibody patterning. I think depending on how you build out the aptamer, like depending on what functional groups you stick on the end of it, you could really exploit that difference in charge and get like a nice self-assembling aptamer brush on the surface. Yeah, I think you might have to change the chemistry a bit. My only experience with aptamers for EV capture so far has been using like gold thiol binding, but you could switch out chemistries and assuming it's a good aptamer, you could change the functional group on the end and bind them pretty efficiently. So I don't think there's any reason why you couldn't use that approach. We haven't done it yet though with this technology.
All right, I don't see any more questions, so I'd just like to thank Colin again for your talk and for taking the time to talk about your really interesting research today. So yeah, thank you. And this talk was recorded, so it will be uploaded eventually to the YouTube channel. So thanks everyone for coming, and we'll see you next time.
Thank you.